Dear students, ladies and gentlemen, I welcome you very much to our next lecture, Scientific Presentations in Biology. You know that this time we are dealing with microbiology, a very interesting topic, but before just starting with it, let's stay a little bit with scientific presentations in general. So we were thinking that you sit in the audience and you don't like what you heard. So this might be the situation where you would like to say something like that. I can't help feeling that. It was one possibility. It is open to question whether, and our next things are a bit more standing. I take your point, but you can see this over there. I see what you mean, but, or I know what you mean, but. So if you're not satisfied at all, you might just say something like that. I take your point, but I see what you mean, but, or I know what you mean, but. So with that, I would like to skip our general vocabulary, and I would like to jump to microbiology. You see this over there. So we were dealing with a few topics already. Botany and plant science. We are specialists in cell biology. We were dealing with development, molecular biology, zoology. Last time we were working with ecology, and now we would like to start with microbiology. You see it over there. So our today's topic is microbiology. Let me start with some general vocabulary before just jumping into depth. So it's of course a lot, and you know that I would like to read this in English, followed by German translation. So I think I will start now. So, vocabulary that's important in microbiology. For instance, adhesion means adhesion. Algae and algae, familiar to us with plant biology, means algae and algen. In microbiology, we have to deal with antibodies. Antibody, what do you prefer? Das Antigen ist Antigen auf English. So microbiology says that we are dealing with a lot of bacteria, for instance with archibacteria. We would like to work with Anhaftung, attachment, bacterium, bacteria in itself, bacterium, bacterien. So when going into depth, we would like to work with cilium and cilia. You know that this is for movement. Cilium and cilien. Bacteria might have a circular chromosome, a zirkuläres Chromosom. They may colonize something, besiedeln. Composition, die Zusammensetzung, and dormant means dormant. Before continuing with that, I would like to explain a little bit on microscopy in itself, because all these bacteria, we cannot see them by eye. We need microscopy to watch them. This is a picture of light microscopy. You see this over there. And I would like to explain a little bit in depth how this is working. You know that you need a subject, something you would like to see. Objective and ocular are standing in the microscope. Objective and ocular. Your eye is watching this. And you may just come up with a picture of there. This is enlarged. You see the subject, but you see it in an enlarged way. So I am a plant biologist. You see this over here. These are cross-sections, reliefs of Arabidopsis. And this is of course done by light microscopy. It is stained. You see this over here. And you see this enlarged way. Whereas when doing without microscope, you would see this in a small version. You see this in an enlarged version by light microscopy. So cross-section means you see, knowing it's a plant biology and cell biology, the epidermis. You see the vascular bundles over there. You see it is surrounded by mesophyll cells. And you may watch on the right version, and this is a mutant, and you see as well the epidermis. You see vascular bundles. The lower epidermis is over there, and vascular bundles are surrounded by mesophyll cells. You can see this here. For us, interesting, it is stained, and you can watch this by light microscopy. Stained, by the way, with stridine blue. It's making its tissue blue. So let us repeat this. This is, by the way, bacterium. You see this by light microscopy over there as well. So I know that we know this, but it's very important. Let me repeat this. We learned adhesion, adhesion. We are familiar with algae and algae. We learned antibody and antigen. Archaebacteria, Archaebakterien are familiar to us. Attachment was die Anhaftung of bacteria somewhere. We are familiar with bacterium, bacteria. We know that psyllium and cilia, one and several, are important for movement of bacteria. We learned it's a circular chromosome. We are familiar with to colonize, composition and dormant. So that's very important, and I suggest we continue with more vocabulary. You can see this over here. Of course, we need to learn this as well. Electron micrograph. Electron micrograph means in German elektronenmikroskopische Aufnahme, another type of microscope. Electron microscopy, die Elektronenmikroskopie. We will jump this later. Endospore means endospore. Eukaryotic, eukaryotisch. Exocytosis is in German die Exozytose, and flagella is in German flagelle. Fungus and fungi, pills and pilze, may be a subject of microbiology as well. fungus and fungi. Gastrointestinal tract. Human health, die menschliche Gesundheit. Infectious diseases, Infektionskrankheiten, may be induced by bacteria. So that's why it's just coming here. Layer, die Lage, and Mikrobe. So, of course, we need more vocabulary, and you see a few words over here. You know that microscopic is mikroskopisch, standing here. The organelle is die Organelle in German. Phagocytosis or phagocytosis, both pronunciations are possible. It's in German die Phagocytose, followed by panocytosis, pinocytose, over there, and very important, Plasmid ist das Plasmid auf Deutsch. So, other words are visible over there, and I think they are not so difficult for us, but the pronunciation is just different than in German. Prokaryotic is in German prokaryotisch. Protozoa or protozoa, Protozoen. Resistant. Resistance ist die Resistenz. Secrete or secretion, Ausscheiden and Ausscheidung. So, with that, I think we learned a lot of vocabulary already, and we use the time in between to start with our text. And of course, it is very, very interesting. I think I read the first 3 sentences over there, and it's going on the halopathogenic bacterium Yersinia pestis. So, for that, I think the text will answer this question. Yersinia pestis, formerly Pasteurella pestis, is a gram-negative rod-shaped bacterial species that belongs to the family of Enterobacteriaceae. Infection of humans with wild pastors may cause the extremely aggressive diseases of pneumonic, septicemic and bubonic plagues. These 3 forms of plague caused the deaths of millions of people throughout human history. For example, the epidemic called the Black Death, which was a form of bubonic plague, accounted for the death of 1/3 of the European population in the 14th century. So, that's of course very, very interesting. A lot of words shown in blue are very important or might be unknown. And I think I will deal with that in the next slide. And this will answer the question, what is Yersinia pestis? Vocabulary for the text. Pathogenic, pathogen. Bacterium or bacteria, So we learned that Yersinia is a pathogenic bacterium. It is gram-negative. You remember our microbiology in the studies, gram-positive, gram-negative. It's a rod. Rod means Stäbchen. Shaped, gestaltet, geformt. You can combine this rod shape, means gestaltet wie ein Stäbchen. Species, art, you remember that from other lectures already. Our Yersinia is responsible for infections, infection, human, oder menschlich. It's inducing disease, Krankheit wird initiiert, und zwar pneumonic plague, Lungenpest, septicemic plague, septichemische Pest, oder bubonic plague, Beulenpest, Bubonenpest. So, let us continue here. The plague or plague ist die Pest, kann auch einfach als Seuche eingesetzt werden. Death ist der Tod. You probably know that. Epidemie oder Seuche. Black Death, der schwarze Tod. Population, Population, Bevölkerung. So, let me continue with reading this in English once more. Pathogenic, pathogen, bacterium or bacteria, gram-negative, war gram-negativ, rod, das Stäbchen, shaped, gestaltet oder geformt, species, infection, human, disease. So, very important words are coming here. Pneumonic plague, die Lungenpest, septicemic plague, and bubonic plague, Beulenpest. You can read this over there. Plague or plague, Pest oder Seuche. Death, der Tod, Epidemic, eigentlich epidemisch, Epidemie wäre Epidemie in German, Black Death, der schwarze Tod, and population, Bevölkerung. So, let us continue with reading. Here it comes. Yersinia pestis, or Y pestis, is transmitted by fleas to its mammalian hosts, which include humans and marines. The bacteria infect the digestive tract of fleas. They are transmitted to mammalian hosts by flea bites, after which they may be taken up by blood-brain fleas again. Interestingly, different species of fleas vary in their ability to transmit viral pests. The red flea, Xenopsula hyopius, is highly efficient in transmitting the bacterium, with the cat flea, Ctenocephalides felis, for example, cannot function as a vector for viral pests. So, I think we learned a lot on plague itself, and we should read the blue words again. Mammalian means Säugetier, Host, der Wirt. Mammalian host ist, Säugetierwirt. Interesting are marines, Marine als Einzahl, mausartig zur Mausfamilie gehörend. Flea ist der Floh, staying over there. A flea bite Sie können es zusammen oder zusammenschreiben, ist auf jeden Fall der Flohbiss. Blood feeding, sich vom Blut ernähren. And we are dealing with rat flea, sitting over there, der Rattenfloh, und cat flea, der Katzenfloh. So, let me read this once more. Säugetier was mammalian, host marine, mausartig oder zur Mausfamilie gehörend, flea, der Floh, flea bite, der Flohbiss, blood feeding, sich vom Blut ernährend, rat flea and cat flea, Ratten- und Katzenfloh. So, we learned a lot of words, but of course this is not sufficient. We need to learn more in microbiology. Here coming a few words next to the text that are important in microbiology as well. Layer, die Lage. Microbe, Mikrobe, so we were talking up now in microscopic, mikroskopisch. Organelle, organell. Pathogenic means pathogen. And we are already specialists in phagocytosis, Phagozytose, and pinocytosis. Plasmid, plasmid. Prokaryotic, meant prokaryotisch. Protozoa waren die Protozoen. Resistant and resistance, resistant or resistance are very important words in microbiology as well. So, this is another type of microscopy, and of course we have to read this. The name is confocal laser scanning microscopy, and of course it is more complicated. You are starting with the subject as well, and you have an objective in the microscope, but in addition you have a scan unit and a laser. You can select the wavelengths, the Wellenlänge, what you are interested in, and usually the wavelengths are just corresponding to the flow force. So in these pictures you see the GFP, the green fluorescence microscope. GFP means green fluorescence protein. You have a laser detecting the green over there. You realize the fluorescence. GFP, green fluorescence protein, and you will ending with a picture. So the charming is that you can use optical sections in confocal laser scanning microscopy. You don't have to cut this in thinnest micrometers. The confocal laser scanning microscope can do this in itself. So these are two pictures of mesembrandum, the ice plant, and you see this GFP over there. You see it. Oh no, I know that this is a control. This is a stress plant, and the GFP is induced. You see this mesofilter as a tiny signal over here. You see strong signals over there. You see the vesicular bundle. It's coming control plants here, and stress plant over there. And red means autofluorescence of the xylem. You can see this here and there. These are two very, very charming images taken by confocal scanning microscopy. You can see this over here, and I think this is the second type of microscope that we learned. We are already familiar with the light microscope. CLSM is coming here. We are familiar with that as well. So, with that I would like to skip our excursion to microscopy, and of course we have exercises to deal with when reading our text. This is the first exercise, and I think I'll read this. Understanding and presenting a scientific article. So we are asked to read the text. It's coming during the next minutes. We are asked to understand this, of course, and to repeat this in talking. We are allowed keywords up to 15, and we are asked to select English keywords. We are asked to read the journal text several times, 3 times actually, and then put this away, and it is meant that we are talking on the text using our keywords, but without watching on our text as well. So I'm fascinated. This is flea and flow. You see this over there, and it's just observed by microscopy. It's so tiny that you probably don't see this, and it looks gorgeous, I think. So, with that I would like to continue reading, and we learn more on Yersinia pestis. I think I read this first. Yersinia pestis influences and interrupts the metabolism of the infected host on different levels. The bacteria prevent the immune response of the host by paralyzing phagocytic cells. In addition, they have the ability to suppress and disrupt infection, induce signal destruction in host cells. Pestis uses human and marine erythrocytes as an essential source for iron acquisition, thereby damaging the erythrocytes. So this sounds very interesting to us, and I think we jump to the words that might be unknown. To influence is not restricted to microbiology, but used here as well. To influence, beeinflussen. To interrupt means unterbrechen. Metabolism, Metabolismus. To infect means infizieren. Level, die Ebene. And to prevent, verhindern. So, with watching our fleas as well, I have to stop at that point. I would like to say tschüss to the next lecture, and I'm looking forward to microbiology and to meet you again.